name is Vikram Narayanan Dhamu. I'm going to be presenting an electrochemical teardown of a trace pesticide assessment system. So the platform we've developed, we've dubbed as Electrochem Sense, which is a real-time rapid pesticide assessment system. The goal is to understand the pesticide contamination in the food we consume. The reason for this being that the health risks for humans due to the exposure and ingestion of pesticides is highlighted in this schematic diagram here. Our solution is to come up with a sensing ecosystem that uses runoff water from produce when washing to test for the pesticide traces. And therein lies the challenge to design a low volume sensor that can screen for glyphosate directly from produce runoff and report it as a useful concentration information. The objectives in this project involves introducing a sensor region into the runoff water sample from the vegetables or fruits. Then this is interfaced with an electronic reader that is portable and which runs the electrochemical tests and thereby computes the interactions between the pesticide analyte and the receptor which we have functionalized on the sensor chip. Then we have a back-end step of a machine learning assessment system that is leveraged to compute and predict the pesticide residue state of the produce, which is highlighted below. So pesticide residue state here means that we, based on a MRL, which is the maximum residue level, we can classify a produce sample as either safe or unsafe within those limits. The MRL is given by different countries and different environmental groups worldwide. So as an example, I've shown it for apple and carrot as 0.2 parts per million and 5 parts per million, which is based on the USDA and the global MRL database. The sensor chip that we've built is the form factor of a USB flash drive for slot in and slot out with the USB receptor or the reader module that we've built. The EIS modality that we apply here is one of our first electrochemical systems that we have tested for this pesticide ecosystem. So wherein we apply a AC bias, which is an alternating current bias, to the electrode that perturbs the interface at the electrode-electrolyte surface, causing a dielectric or a capacitive effect. This impedance or a capacitance is monitored as a function of binding between the pesticide analyte and the receptor ligand, which is the antibody, which is shown in the schematic right here. So in a more robust or a fast mechanism, we used chronoamperometry, which is highlighted in the left here, where we actually use a DC step signal that is being applied as an input, and the current trend is measured against time, and interfacial current at the peak due to the binding effect is tabulated. This is measured as a function of the potential difference applied between the reference and the working electrodes. That is actually simulated in COMSOL, which is a finite element analysis software, and that actually shows on the right here that when we actually keep one electrode at a standard reference point or a grounded state versus applying a bias at the other electrode, we see a clear potential difference that is actually causing our electrochemical bias and electrochemical modulating effects, which is the driver of all of these signals. Moving into the results, we see that the electrochemical impedance spectroscopy results, which is in the form of a Nyquist or a Bode plot. First, we can talk about the Nyquist, where we have the real versus the imaginary portion of impedance, and the bending of the signal with respect to dose shows that the binding effect is actually being tabulated in terms of the impedance. Next, we move on to the Bode plot, which is measured as a function of the magnitude impedance and the phase impedance, where with more binding, which is modulating the impedance, there's a signal change, and that is concentrated, that is shown as a recovered concentration trend here in the form of the bar plot trends in both the magnitude and the phase curves. In terms of the chronoamperometry signal, we have the peak current, which is modulated as a function of the binding, which we call as the capacitive current or the interfacial current, which is measured against time, from where we basically observe and tabulate the min-max current obtained at 0.01 seconds or in the negative phase at 15.01 seconds. So then we actually plot this current against the dose to see a trend. When we see a linearly changing trend, we can see that the binding modulation is causing the signal change corresponding to the recovered concentration. Next, to actually prove the temporal incubation time factor or the temporal effect of binding, we've actually observed and tabulated this in terms of 5 minutes, 10 minutes, and 15 minutes incubation time. So when we see that the 5-minute incubation of the analyte and the receptor ligand binding shows a best linearity in terms of a 5-parameter plot. So therein we have concluded that the rapid response time of 5 minutes is possible by this electrochem-sense system. To conclude, we have a limit of detection with the lowest distinguishable range as 1 parts per billion and a field operating range which is fine-tuned for each produced item for the best performances, and we've characterized and tabulated this as well. Also, we have the machine learning algorithm at the back end for optimization of the performances, which shows greater than 75% accuracy for complex field produced samples. The confusion matrix exactly depicts these results right here. We have published more than 2 papers with more coming on this work, but further work is still needed for in-field market testing and optimization of the ML system. We would like to thank you all for listening, and I can answer any questions you may have now.